The automated MeshAndCollect protocol was developed to combine serial crystallography with standard rotation data collection to measure small crystals or from opaque samples.
The method first identifies the position of crystals mounted on the same sample holder, to then direct the collection of partial dataset that will be subsequently merged and used for the solution of the structure.
MeshAndCollect is compelling as it allows you to do experiments rapidly and with the same setup. Structure solution, ligand screening, and all that with tiny crystals.
The method is compatible with synchrotron beamlines for MX, ideally equipped with high photon flux, a small beam diameter, and a fast readout detector.
First, connect to the Extended Information System for Protein Crystallography Beamlines database and choose MX. Log in with the experiment number and the password from the A form. Next, select Shipment Add New and fill in the requested information. Select Add Parcel and fill in the relevant data. Select Add Container, choose an SC3 puck, and fill in the information required, including the positions of the sample holders in the puck. In the experimental hutch, load the puck into the sample changer dewar and note its position. Next, log into the information system for Protein Crystallography Beamlines database. Select Prepare Experiment, find the shipment, select Next, and indicate the beamline and puck position in the sample changer. Log into the beamline control software with the experiment.